Hello everyone, and welcome to the last video lecture of the semester. Today we're going to cover Chapter 14, Replicability, Generalization, and the Real World. So we're going to look at bringing our science into a larger environment and what it means to do so. So we're going to talk about questions of replicability. So for a study to be important, it must be replicable. Can you replicate what you have found in a different study? And then we're going to address the question of, for a study to be important, must it also have external validity? And we'll get into quite a few points on that one as well, but that's our outline for today. So for our replication studies, we can talk about three types of replication. We can have direct replication, conceptual replication, and replication plus extension. So we're going to go into examples of each of these to help illustrate why we might use any of these types of replication to look at research. So with direct replication, which is sometimes called exact replication, the original study is repeated as closely as possible. So you conduct a follow-up experiment that is the exact same experiment over again. And you want to determine if the original effect is found using new data. So using the same experimental setup, the same kinds of questions, the same design, the same everything as possible, if you have a new group of participants, do you find the same effect? So do you get a direct replication of that original study? And so we can talk about an example for this, where Jones and colleagues back in 2004, they theorized that most people like themselves. That's a pretty well-known fact for psychology. People tend to think highly of themselves. But the researchers thought that they might implicitly associate positive feelings with things that they associate with themselves. So things like the letters in their own name or the numbers in their birthday. And so these researchers hypothesized that people might be more attracted to people who share their name, birthday, or initials, so sharing features similar to themselves. And to test this hypothesis, they had participants complete a short survey about themselves. Then each participant would be given the page of responses from a randomly selected partner. And at the top of the page, the partner's ID was printed in large font, so a string of ID numbers. For half the participants, the partner's ID number had specifically been matched to the participant's own birthday. So if you were born on September 25th, then the partner's page that you were given would have the ID number 09-25. So there would be some familiarity and maybe a connection there. For the other half of their participants, there wasn't a link in the participants' numbers and their birthdates. All of the other information on these forms, so even though the participants were told that this is this other person's information from the survey, they held that information constant. So everybody saw the exact same information on the page. The only thing that differed was the ID number at the top that either matched the participant's birthday or not. So after giving the participants time to read over the surveys, they were asked to rate their partner on a bunch of different dimensions. So here we're looking at overall liking. So how much do you like this random person based on some very simple information on a form and whether or not their ID number matches your birthday. And so in the first study, 5A here, we're looking at our control group, so no matching, versus the experimental group with the birthday match. And we can see that our birthday match group had a much higher report of liking than the control group. And so here, they just did a direct replication. They ran the study again a little bit later in time, same exact setup, and so they called this 5B, and even though the bars aren't exactly the same heights, we're still seeing the same pattern. We're still seeing the overall effect here. So we would call this a direct replication. And that gives us more confidence in those original results because we found the result again. Though, of course, there are always things to consider. So even though this direct replication method makes some conceptual sense, when using one, you still run the risk of repeating any threats, any problems that you hadn't thought of in the first study, and they pop up again in the second study. So any threats to internal validity, any problems with construct validity in that original study are also going to be problems in your replication study. So sometimes researchers have to take some other version of replication instead. Direct replication is not always the best option. So what about our next option, which is conceptual replication? So for conceptual replication, researchers want to explore the same research question, but they're going to do so using different procedures. So the conceptual variables are the same. It's the same basic idea of what they're looking at, but the procedures they use for operationalization is going to be different. So they're going to have different operationalized variables, though the conceptual levels will be the same. And so we can keep going with that idea that people like things about themselves. Or we can actually call it what it is, which is a theory of implicit egotism. So that built-in self-importance. So the last study had looked at sort of an independent variable of similarity to the self, so having a similar birthday or an association with a birthday. And then they had tested it using their dependent variable of attraction to a person or liking. That was in the last study. So we have this similarity to the self as the IV and social behavior as a DV. And so we saw that people who had ID numbers similar to your birthday, you end up liking them more. But if we wanted to conceptually replicate that idea, if we wanted to show that that same concept applies in multiple ways, we could generalize. And so for some quick examples here, in conducting some other studies, researchers have found that people named Dennis or Denise tend to be dentists more often than they are lawyers, because dentist sounds similar. But people named Laura or Lawrence are more likely to become lawyers than dentists. Now that isn't so much an experimental setup because you're not determining their name or their profession, but that would be a correlation that adds to this idea. So it could be a conceptual replication because we're looking at similarity to the self and some kind of output social behavior. So similarity to their first name and the career they pursue. And this is something in psychology that has been studied from tons of different angles. People are slightly more likely to marry someone whose name is similar to their own than would be expected by chance. There's picking professions that are close to your name, liking people that have things that are similar to you. There's tons of ways that this has been conceptually replicated, which is kind of cool. For our last type of replication, we can look at replication plus extension. So this is where researchers replicate the original study, but add some variables to test additional questions. So if we think back to our note-taking example, where they had people listen to a TED Talk and take notes on either a notepad, so by hand, or on a computer by typing, and then they tested how their retention of the information was. So they had looked at how do they answer factual questions versus how do they answer conceptual questions. And we had found that the longhand notes were beneficial for conceptual questions. So the people who took handwritten notes answered more conceptual questions correctly. So that was the original study, and you could do a direct replication of that study, but the researchers had noticed that people using laptops were taking verbatim notes. So they wondered if that verbatim copying was part of the problem. So instead of a straight replication, they did a replication plus extension, and they had their original two groups, laptop notes and handwritten notes, but they introduced a third group where they had students who would take notes with the laptop, but they were specifically informed not to take verbatim notes. They said, make sure that you put the notes in your own words. And so when they evaluated this replication plus extension, they actually found that the laptop notes, when they were warned not to copy, performed fairly similar to the people who had laptop notes and no extra instruction. The people warned not to copy still had pretty much verbatim notes, and they ended up performing more poorly on conceptual questions. So laptop notes with no additional instructions and laptop notes told not to copy both scored worse than people with longhand notes, who tended to score better on those conceptual questions. And another way that the researchers had done this replication plus extension was actually adjusting how the students were tested. So for this graph over here, they had looked at students who took laptop notes versus students who took longhand notes, and some of them were given the opportunity to study those notes a week later and then take a test after that week had elapsed, whereas the other group didn't have a chance to study. So this kind of sounds like a 2x2 factorial design, if we're still thinking about those. But here they found that people who took longhand notes and had a chance to study did much better on that test 1 week later, whereas everybody else tended to do less good. So these longhand notes are better in this situation that kind of matches real life a little bit better. So in classes, when you're listening to a lecture and taking notes, you tend not to be immediately tested. You tend to be tested at some later point in time. So this gives them that extension where it's a little bit more applicable to the real world. So all of those examples and some conceptual thinking can get us agreeing that replication is probably a good thing. It's going to have us more confident in our results, maybe help us be more confident in generalizing our results, all good things. Now, in reality, there's somewhat of a replication debate, and our textbook focuses on this replication debate in psychology, but this isn't a problem exclusive to psychology. So we're going to start with a little bit of background on what they're calling the replication crisis. And then we can talk about some of the specifics about why replication studies can fail and what we can do to counteract some of this stuff. So a little bit of history here. So one of the big problems that we have, and this is nothing new, we've kind of touched on this already, is that journals prefer to publish new methods and new theories. They want something new and exciting. And that's not just scientific journals, that's also the public media, but that causes a problem because replication studies are neither new for methods or theories. They're exact copies or copies with modification of things that have already been shown. And so there's this bias in publication where replication studies are not published as often. And the problem with trying to get published or studies published when they're replications led a group of individuals who called themselves the Open Science Collaboration, or the OSC. It led them to start a systematic investigation of replication. So they wanted to look at a bunch of studies that had been conducted and see if those studies held up to replication, if they could get the same results from running those studies themselves. And so this group selected 100 studies from 3 of the major psychology journals, and they contacted a bunch of researchers around the world and asked them to try and replicate the results. And they specifically went with direct replication. So following the methods, following everything as closely as possible, do you find the same result? And so they used a bunch of different metrics. They used a bunch of different measures to try and quantify what these replication studies showed. And by one metric, they reported only about 39% of the studies clearly replicated the original effect. So that's a 39% success rate at replication. That's a pretty low number, especially if we are looking at science and things published in scientific journals and peer-reviewed as things that we have trust in. It's important here to note that this was only one of the metrics that they were using. They had other measures that had much better rates of replication, and still not 100%, but not as bad as 39%. But as soon as the media got hold of these numbers, they reported the 39% because that is the most astonishing, the most alarming thing that they could report. So it's good to keep that in mind as well, but still, having not high levels of replication here is alarming for scientists and for people who consume science. And this is where our replication crisis was born. So after this happened, people were desperate to figure out what was going on and what was wrong with science that they couldn't replicate the results. And so when people are critically evaluating this setup, we can actually look at some of the themes that they pointed out, all summarized very nicely in this table from the textbook. But we can look at problems with the replication attempt. So was there something wrong with their direct replication? Maybe there was a problem with the original study, or maybe it's something that's wrong with the system of publishing academic articles. So we can look at some of these potential problems, just very briefly summarized here. There's more detail in the text. But with our replication attempt, so replication tried to use exact operationalizations. Always miss a syllable in there. Operationalizations from the original study, but sometimes they weren't relevant in the replication setting. So if you had conducted a study in North America and it was relevant to North American ideals, if the people replicating the study were doing so in another country, maybe in a culture that has different ideals or views, then the thing that was being manipulated in the original study might not be manipulated in the same way in the replication attempt. So you could have your replication study fails because it wasn't really a true test of that original effect. So in order to look at that, you kind of have to critically think about what was being replicated, and was it specific to the original population, and is the replication population close enough to have worked? You can also have the issue where the replication didn't use a large enough sample. So if the original study used 400 undergraduate students, but the replication only used 20, maybe you didn't have a large enough sample size to find the effect. So you concluded null results simply because you didn't have a large enough sample size to detect the effect. So again, some critical thinking and evaluating the design of that replication can help you with that. For problems with the original study, maybe it was the original study whose sample size was small. And when we first talked about sample sizes and finding effects, we had mentioned that small sample sizes, so studies with very few participants, are prone to being influenced dramatically by outliers, by extreme instances that are reported, usually just due to chance. So if you have a small sample size, then maybe you have a high chance of the thing that you found isn't actually a real effect. In fact, it's just an artifact of some kind of random occurrence within your data. So larger sample sizes, you have more people there, and so even if by chance you get someone who scores really high on your dependent variable, if it's because of chance and not your experimental manipulation, there's also a chance that someone scored really low on that DV, and those individual variations, all of that noise, can cancel out. So in that case, with the original study being the problem, then the original study should focus on having a sample size that's large enough to be more certain that what you've detected is a real effect. We can also have problems where the original study used some questionable research practices. So maybe they ran an experiment with lots and lots of dependent variables. Maybe you were looking at whether money increases happiness, but you also measured life satisfaction and depression scores and 12 other things. But only one of those 12 had a statistically significant relationship with income. Some researchers would only report one of those things, the thing that was significant. They might hand wave all the other dependent variables that they were measuring. So by kind of picking and choosing things, you're missing out on the big picture, and you're only reporting something that makes a splashy headline. For problems with the publication process overall, this is stuff that we've already kind of talked about, where scientific journals prefer to publish new stuff over replications, but also that journals tend to prefer to publish a significant finding over a null effect. So we have this double whammy where the publication process is fairly flawed, and we have to keep that in mind when looking at the literature. Now, for some more specifics and the correct terms for some of the general ideas that I was discussing with that chart, we can sort of break it down this way. So at the beginning, when I talked about the fact that in different cultures or different countries, the thing that was being manipulated might not be the same. Maybe they have different core values, or maybe the manipulations are acting differently because of cultural perceptions or something like that. We can talk about those effects that are or are not happening being contextually sensitive effects. So some of our measures and some of our manipulations might not have the same meanings in a replication population as they did in the original sample size, or the original sample that was actually a good example of this. The textbook talks about how an original study was looking at how people contributed to charities and sort of what percentage of people would contribute to a charity. And in the original study, they mailed out actual physical letters, and it was a charity about AIDS and raising awareness. But in the replication study, the researchers sent out emails instead of physical letters. And the emails were about an environmental charity, not an AIDS sponsoring charity. So there are a bunch of different things that weren't quite the same there. So maybe the failure to replicate had something to do with the fact that emails are less tangible than mail, or that people would respond to an AIDS charity more than an environmental charity, or vice versa. So there's a bunch of different ways that the original context can be lost, and that might be what's driving this inability to replicate. For our number of replication attempts, this is an interesting one from a statistical side of things. So when you conduct your original study, maybe you find a significant result, but now you're not sure if that's a real result or if it was maybe due to chance. Maybe there isn't really a real effect, but you found an accidental effect that's just an artifact of your data. So you conduct these replications. If your replication study, maybe there's only one person that tries to replicate your results. If they don't support your results, now you have two people, one who says that it is there, one who says that it isn't. Which one do you give more credit to? Is it more likely that there was an effect and the replication study couldn't detect it? Or is it more likely that there wasn't an effect and by chance you accidentally found an artifact that looks like an effect? It's hard to say which is correct with only one original study and one replication. So one replication attempt does not necessarily tell you anything important. And then for our last thing is problems with the original study. I've already kind of talked about these problems, but I didn't have the names on the previous slide because I forgot I included them. So the sample size problem was pretty simple to cover, where having too small of a sample size in the original study can mess with your data because of chance variation. We can also run into problems with how researchers are looking at their results. The first of these problems is called Harking, and the HARK in it is actually an acronym for hypothesizing after the results are known. So sometimes researchers will create a hypothesis to explain the results that they got after the fact. So they would sort of reevaluate what they were looking at and come up with a solution, a hypothesis that makes sense in the light of the results that they got. So the problem with Harking is that when it's presented in a journal, if the scientists aren't clear and transparent about the fact that this hypothesis came after the data, then the people reading it might just assume that this is what the researchers were looking for, which isn't always the case. Another problem that can be run into is called p-hacking. So if a researcher has run a study and they're checking their statistics and they find that instead of a p of 0.05, maybe they have a p of like 0.055. So just a little bit above that cutoff for significance. And so if they're convinced that there's something there, but they didn't quite run enough people, they don't have a large enough sample to detect this effect, then maybe they run some extra individuals to try and get a significant result. They want to sort of bias their outcome to have that significant result so that they can publish it. So running more individuals is an example of this. Maybe they want to start removing a couple of outliers because it makes their effect stronger. Or maybe they're going to run a different analysis. Maybe they're going to run a less strict look at their data. So when they are p-hacking, they are adjusting or playing with things that are affecting their p-value to try and get a significant result. So obviously not the best choice to run good science. So these are things that you have to watch out for. And it's starting to become more of a thing where the journals are strict about proposing things up front sometimes, but also a good researcher should know that these aren't good practices. And so these things have to be disclosed. So a little bit of trust on the side of the original researchers. So if these are the problems, how do we solve them? Larger sample sizes in both the original studies and replication studies would be fantastic. There's been a push lately to report all analyses and all variables. So if you run a t-test first and then decide that your results look better if you run a regression, you would still report that t-test because you ran it. For the idea of variables, even if you had multiple dependent variables and only one was significantly associated with your IV, you would still report all of the DVs that you measured. So you are being transparent and open about everything that you ran. Another new practice falls under the term of open science collaboration. And this is the idea that a lot of researchers now are opening up their data and their materials so that others can actually look at exactly what they did. So if you open up your data, then another researcher could come in and run their own analyses using the exact data that you generated. So they could replicate your findings by looking at the same data. By opening up your materials, say you ran a survey and you released the materials that you used to run the survey, people could replicate your findings using the exact same survey questions that you had used, and they could exactly follow your methods. So this collaboration has been a big drive lately to get people to be more open about what they're running and how they're running it. And then an interesting one that actually, this was new to me because I haven't encountered this in my side of the field. But this is something called pre-registration, where some scientific journals are encouraging scientists to pre-register their studies, methods, hypotheses, or their statistical analyses. So before you conduct a study, you would reach out to a journal and tell them your intent to study something. And in some cases, you can even have that pre-registration peer-reviewed, so people could look at your proposed methods, your ideas behind it, and the statistics you would run, and they could critically analyze things that you should also consider. So a peer could suggest that you include an extra measure to account for a potential third variable. Or somebody could propose a better statistical analysis that could help you look at your data in a better way. And because you would register in advance, then there isn't any worry of p-hacking or harking because you've already said what you're going to do and how you're going to do it. This also comes with some extra bonuses because sometimes journals will commit to publish your research no matter what kind of finding you have, which could get you the opportunity to publish a null result if that's what you end up finding. So some interesting improvements. This one must be fairly new because, like I said, I haven't actually heard of this before, and I will definitely be looking into it because I really like that idea. So our next couple of slides are going to touch on meta-analysis, which we've already kind of talked about meta-analysis way back at the beginning of the course, where we talked about sort of conducting a statistical analysis that's going to give us a quantitative summary of the scientific literature. And what I mean by the literature, I mean all of the studies that are related to a particular concept. So if we talk about the literature around income and happiness, then we would be talking about all of the studies that have been conducted by all of the different researchers who are looking at happiness and income. So your meta-analysis could let you look at all of those studies and all of their results in a condensed fashion to give you effectively a larger sample size because you're condensing across multiple studies with lots of different participants. So we're going to look at this using an example and then talk about some of the strengths and limitations that come with meta-analysis. And so this is going to be mostly logic and not so much new information, but that's okay. So the example here is the idea that lying takes time. So in the popular media, when you're watching TV shows, they always show criminals having a lie detector test, and there's some kind of physiological stress response associated with lying. In actuality, there's not so much a predictable physiological response. It's not as clean cut as the TV shows would like us to believe. And in fact, a polygraph test's output is not submissible in court because it isn't necessarily proof of guilt. So there is another theory about lying and what's going on, and this theory says that lying isn't so much a physiological stressor, but it's a cognitive stressor. So in order to tell a lie, you have to think hard about what lie you're going to tell, you have to make up that information, and you need to keep your story straight. You need to make sure that you're not contradicting yourself. And so all of this is taking up mental processing power. And the idea is that if you are making up a lie, it should take you longer to respond. And so in this case, there are a bunch of studies that have looked at the amount of time it takes for people to respond to an object. And if they're lying in their response, they overall tend to take longer to respond. So, for example, people would be given an envelope, and in the envelope there would be a $20 bill and a jack of spades. And their instructions are that you are supposed to lie about having seen these items. If asked, you've never encountered these before. And so then the participants would sit down at a computer screen and they'd be shown different playing cards and different bills and asked, have you ever seen this before? And so the idea being that they would take longer to say no when shown the jack of spades or when they were shown a $20 bill than any other card or any other bill, because in those cases they were lying. And so a bunch of researchers back in 2017, they decided that they were going to evaluate the full literature on these kinds of lying experiments. So they collected 114 different experiments that used a very similar setup. So they computed an effect size. In this case, they were using D, Cohen's effect size. And they calculated a D for all of the experiments. And in this case, a larger D would mean that they had a larger difference in reaction time between lies and the truth. So a large D means that they took longer to lie and they were much faster telling the truth. So they were looking at the difference in lying and truth telling for response time. So they calculated all of those effect sizes, and then they could look at the overall effect of the literature. So by looking at all of these different studies, what are they finding? And in fact, they ended up finding that they had, in a lot of these cases, very high Ds. So if we remember our Cohen's Guide for Effect Sizes, we've talked mainly about R for this course, and the people conducting this meta-analysis could actually have used R if they wanted. They just used D. It doesn't really matter. But pretty much every number that we're seeing is a very large or strong effect size. So 0.8 is large or strong, and we're seeing up to 0.97, 1.29, even higher over here. So very, very large effect sizes. And they could even use the data they had collected to break things down. So in this graph, they're showing the difference between studies that had instructed the participants to go as fast as possible, who were told to respond quickly, versus not told to go fast. And being told to go fast meant that there was a larger difference between their truthful responding and their lying responding. They also looked at what happens if people were motivated to lie. So if there was no motivation to lie, they had a very high D still. But if they were told to avoid detection, if they were told to be aware and not let on that it's a lie, that number decreases. And that decrease also holds true if they had an incentive to avoid detection. Maybe they were monetarily motivated. These numbers are still smaller than when there was no motivation to lie. And the reason why that matters is because the people are, when they're being careful not to lie, the difference between a lie and the truth is closer. So when they are motivated, there's less of a difference to observe. I don't think I made that clear when I first said it. So for the strengths and limitations of meta-analysis, the strengths are pretty obvious. By looking at multiple studies, you're effectively looking at a much larger sample size. It can give you really, really large effect sizes because you're looking at such a large and diverse collection of data. So these meta-analyses can give you a good idea of the overall trend, and sometimes when it's really hard to conduct a study with a large enough N to be confident in your results, by condensing across multiple studies, you can kind of get rid of that problem. Now for the limitations, which is what this slide is really all about, we run into some pretty serious problems to consider, but again, they're nothing new. So the textbook calls the first issue the file drawer problem. And this is the idea that a meta-analysis could overestimate the true effect sizes because null effects or even opposite effects, so studies that would have found that there's no difference between lying and telling the truth, or studies that showed that people who were telling the truth took longer to respond, those studies don't necessarily get published and therefore aren't necessarily included in the analysis. So it's called the file drawer problem because null and opposite effects might be kept in the researcher's filing cabinet. They might never submit them for publication, or they might never be accepted for publication, so they're kept in the researcher's stores. So this is coming from that problem with publication, this bias in publication where null results in particular are very difficult to get published, and so you don't tend to see them. So even if you're looking at all of the published research, you aren't necessarily getting the full picture. Maybe you're only seeing the positive results. And so a fantastic example of this is looking at studies of antidepressants and how they improved depressive symptoms. And so in this one meta-analysis, they went the extra mile and actually contacted researchers and asked for all of their data, whether published or unpublished. So they looked at 74 studies. 38 of them showed positive results, 24 showed negative results, and 12 were somewhere in the middle. And then they looked at which ones had been published. All but one of the positive results had been published in medical journals. So 37 of 38 had been published with positive results. For the null and negative findings, they were much, much less likely to be published. So if we collapse those two groups, of the 36 negative or neutral outcomes, only 3 were published. So if the researchers conducting this meta-analysis had only used published studies, then they'd have a 94% agreement that these antidepressants are effective. But when using all studies, published or not, there's a 51% success rate. And that's a huge difference. So that's something to definitely keep in mind. And maybe when conducting these meta-analyses, researchers should be going out of their way to find unpublished data as well. Another thing to consider about replication and popular media is the usual problem where journalists don't always consider the importance of replicability. We talked earlier in the course about how journalists don't report on the entire collection of studies in a particular area. They're going to focus on what is the most recent or what is the most exciting study, but they're not really going to give you a summary of the whole body of literature on that topic. So we run into a very similar problem where if you're using public media as a resource to understand a particular topic, you're not getting the full picture, which has problems in multiple areas, as we've talked about ad nauseum throughout the course. So that's replication. Now we're going to move on to not so much a statement that replication is important, but now a question of must a study have external validity to be considered important? And so we can look at external validity in terms of generalizing to other people or generalizing to other settings, and sort of what it means for our interpretation of that research and its importance in our minds. So again, overview of the next couple of slides. Now, a lot of these points are going to be fairly similar because we're talking about external validity, which we've talked about throughout the whole course. So these shouldn't be new concepts. But when we're looking at generalizing to other participants, we care about how these participants were obtained. How was this sample gotten? So our probability samples should probably generalize to the population they were drawn from, but a convenience sample might not do the same. We also have this issue of it's a population, not the population. So when you are sampling, you're not necessarily trying to represent the entire population of humans in the world. That is not the population of interest. Our population of interest might be much smaller. Maybe you're only conducting research at the University of Alberta, so maybe you're trying to have a population of first-year undergraduate students in Edmonton. So there's a big difference between the population that you're drawing from and where you can interpret that to apply to. So there's a lot of critical thinking that goes involved here in separating out what your population is from how you're sampling versus what you would like to apply your data to. And that's, again, nothing new. We know that there are limitations of how you can apply your data based on the sample you've drawn from. We should also keep in mind that external validity cares more about how you got your population or how you got your sample from the population, as opposed to how many participants are in your sample. So the idea here is I would much rather have a small randomly obtained sample as opposed to a large non-random sample. So I'd rather have 200 people that are representative of my population of interest than I would have 1,000 people who don't represent my population of interest. External validity, we care about how we got that sample and if that is a good representation of the population we want to study. And that leads us again to that last idea that just because a sample comes from a population doesn't mean it generalizes to that population. We still care about how we got that sample from the population. Was it randomly obtained? And specifically, is our sample representative of the population as a whole? So again, critical thinking about who's in our sample and how that relates to our population of interest. In terms of generalizing to other settings, in the case of a lot of research, and specifically experimental research, but pretty much anything that's been conducted in a laboratory setting, we tend to focus on the ecological validity. Which is sort of a subset of external validity where we're caring if a laboratory conducted study would generalize to real world settings. So they can also call this mundane realism, but we're basically asking, does this experiment that was conducted in the lab have any applications in the real world? So if we used our example for lying about having seen a playing card, that study was conducted in a lab setting, very controlled. But does that tell us anything about lying in the real world? Does that same idea hold true for people who are lying to the cops about stealing a pack of gum? That is sort of the ecological validity of that study. So if we're caring about generalizing to many people, to larger populations or different populations, we're going to talk about things like being in theory testing mode versus generalization mode, which is basically, have we prioritized internal validity or external validity? And then we're going to focus in very briefly on cultural psychology, because it's a very special case of generalization mode where they're focusing on the ability to generalize different ideas, specifically across multiple cultures, so therefore multiple populations. And then we're going to look at weird patients and whether that's a problem or not. So let's start with theory testing mode. And this is what the textbook uses to talk about research that's testing a specific association claim or causal claim. and specifically when those studies are set up to find support for a particular theory. So back in chapter 1, we had looked at the theory data cycle. So these researchers in theory testing mode would be using that theory data cycle to try and test a theory and refine their theory based on what kind of evidence they were generating. And in these cases, external validity is far less important to the researcher than internal validity. And a lot of basic research falls under this category. So a lot of psychology is going to be conducting theory testing mode type research where they're focused on that internal validity. They'd rather be certain of what they're testing in this exact setup than they are concerned about generalizing their results to other situations or other populations. We've already talked about this idea using an early on example when we talked about that comfort contact theory. So when Harlow was looking at infant monkeys and the attachment method they were using with these artificial mothers. So his studies were designed specifically to help him differentiate between the cupboard theory, if the babies were attached to this mother because it fed them, or if there was support for the contact comfort theory, where the babies attached to their mothers because they were nurturing and soft and comforting. So the study was specifically set up to differentiate between those two theories. Harlow didn't care that his lab-raised monkeys, these infants who had been deprived of contact with other monkeys, they're not representative of monkeys in the wild. They're not representative of monkeys in general. They were a very specific case, but they allowed him to actually study the theories that he was trying to study. So he accepted the fact that there was very little external validity because he cared about looking at and providing evidence for one theory or the other. Another example from the textbook is the idea of parents acting as your grammar coach. So the idea here is that children come to learn grammar because their parents correct their grammar in conversations. So if your child would say, I are a girl, then you would say, no, you would say, I am, not I are. But when they looked at a particular study, so this was back in the 70s, they quantified a bunch of parents talking to their children as they were learning to speak, and they found that the parents corrected factual inaccuracies a lot more than they corrected grammatical inaccuracies. So if the child was talking about, Mom, he is a girl, the parents tended to say yes because the mother is a girl, but he is wrong grammatically because it should be she. But the parents tended overall to focus on factual accuracy instead of grammatical accuracy. But so that was giving evidence against their theory that parents are grammar coaches. And we can look at this study, and this one in particular was conducted with upper middle class families in Boston, and specifically people who were comfortable with allowing researchers to record their conversations with their children. So they're not really representative of the whole world or even representative of the US on its own. But the researchers were testing this particular theory. And because this theory didn't hold in this situation, they now have more information that maybe there's something else going on. Maybe they'd be better off looking at parents' correct factual information, and maybe grammar is coming from some other method, some other kind of factor driving it. So again, less interested in external validity and more interested in internal validity, specifically as it relates to the theories they're looking at. In terms of generalization mode, this is when researchers are focused on generalizing their findings from a particular sample to a larger population of interest. So researchers can choose to focus on the ability to apply their findings more broadly. So their primary concern here is external validity. So a lot of all frequency claims are basically run in the attempt of being able to generalize. So if you want to be able to say that 25% of American women go to university after high school, then you're going to want to have a fairly decent sample of American women who are leaving high school and going to university or not. So you want to be able to generalize when you make those kinds of claims. In terms of research, we talked about basic research being focused on internal validity. Applied research tends to be more in this generalization mode where they focus on external validity. And if you remember, applied research is when you're trying to solve a particular problem. So you might run a study to try and understand effectiveness of antidepressants, but you'd want to make sure that that effectiveness doesn't just hold true in a laboratory setting. Maybe you want to make sure that it's going to hold true out in the real world as well. And so frequency claims, pretty much always generalization mode. Association and causal claims, sometimes. It's not always the case. And this is, again, where researchers can choose if they want to focus on internal or external validity based on their design and what they want to do with their results. And in a lot of cases, when these kinds of studies want to be able to generalize, they might start with a smaller sample to begin with to get them some evidence that their concept might hold true. So maybe you're going to have a marketing researcher who conducts a focus group or a limited sample to evaluate the effectiveness of a marketing campaign. And so if it works in the small scale, then maybe you want to follow up and conduct a larger study, have more people ask their opinion to see if it applies on a larger scale. Doing a little bit of replication with some extensions there, so that you can see if this would generalize to a larger population. So there are ways to do that, and you're going to see that with both association and causal claims. Now, as promised, we're going to touch very briefly on cultural psychology, where they tend to focus on generalization, but in a very specific way. So cultural psychology is a subset of psychology where they're focusing on how cultural contexts shape how humans think, feel, and behave. So a lot of cultural psychologists focus on generalizability because they want to look at how these different ideas and concepts work across multiple cultures. So they're intentionally trying to look at larger populations. And so a great example of this is what's called the Mueller-Liar illusion, which I didn't know what the name was, but as soon as I saw these two images, I knew exactly what they were talking about. And so the idea here is you would show people line A and line B, and they'd ask which line is longer. And most people, especially in North America, will say that line B is longer. But if you're familiar with this illusion, you know that the two lines are exactly the same length. So this is an illusion because these extra lines on the end, these little arrowheads, give us the illusion that this one is shorter and this one is longer. And so you can probably see where this is going, but for cultural psychologists, they'd want to test, where does this illusion hold true? So in North America, people tend to be dramatically influenced by this illusion. So we're measuring degree of illusion. So how fooled are you versus where in the world you are from. And so in Illinois, people are very illused. They are fooled by this illusion. And most North Americans will have that same thing. Europeans, also fairly subject to this illusion, but if we get away from North American ideals, when we get into other cultures, some cultures aren't fooled by this at all. And the current theory is that what's happening is that people who grow up in what's called a carpentered world, basically meaning that they grow up in a world with lots of right angles and lots of straight lines, we start using these right angles, like the corner of a room, as cues for how close or far away things are. So we would interpret this as a corner. So maybe this is a corner pointing towards us with hallways going away from us. Or maybe this is us facing the corner of a room with walls coming towards us on either side. So based on what we've been exposed to, we would interpret these differently than someone from, say, a country where they don't have a lot of square-built structures. Maybe they're raised with circular rooms, or maybe they live most of their time outside and away from solid architecture. Experience changes their subjective experience of this illusion. Another example is how people take in information. So here we have an image with 3 big fish in a fish tank. If you ask people to describe this situation, again, people in North America will say, oh, there's 3 fish. Oh, there's also some smaller fish in the background. There's also some plants and maybe some frog and snail. But they talk about the fish, the foreground, first. People in Japan will actually talk about the background first. They'll describe the environment before they talk about the foreground figures, the fish. And this ends up having an effect on how we're encoding the information because we're focusing on the images very differently. So we're actually looking, wrong way. So we're looking at, so if you're shown this image, how many people can recognize the fish? So if you're shown the fish on the original background, so this guy here in his original context, if we're shown just the fish with no background, if we're shown the fish with some new novel background, what happens? And so we can compare people from Japan and people from North America, and with the original background, people from Japan have a very high accuracy at recognizing the thing they've already seen. So they encode the information of the fish along with the background. When that original background is removed, people from Japan struggle to recognize the fish on its own. The idea here being that they encoded the fish in context, whereas North Americans just cared about the fish on its own. And then if we put a novel background, we still see that the North Americans perform better, but Japanese individuals struggle now because not only is it the fish in a new context, so it's away from their original background, it's also just the fish, so they didn't encode it separately. So we see a difference in encoding based on what they pay attention to when it's exposed. So again, things are changing across cultures. And then this brings us to the idea of weird participants. And we've kind of, again, talked about this without naming it all throughout the semester. So most research in psychology is conducted on North American college students. So people are Western, educated, industrialized, usually rich, and democratic. So we are focusing on a very specific group of individuals. Sometimes when you're conducting research, if you're just trying to prove a theory, it doesn't necessarily matter that you're using this weird sample because you don't care about generalizability to other populations. But if we're looking at that cultural psychology where we're seeing major differences in perception and encoding of information, if you want to be able to apply your research more broadly, you have to be testing in other areas. You can't just look at people from North America, Europe, Australia, or Israel. You have to focus on other places as well. So just sometimes it's okay but other times you really need to be aware of what your sample is, what population you're looking at, and where you want to generalize. All right, and then for the last couple of slides, we're going to look at the idea of if a study has to take place in a real-world setting to be a valid study. So we're going to look at external validity and the real world, generalization mode and the real world, and theory testing mode and the real world. And this is kind of important to talk about because most people assume that studies conducted in the real world are more important than studies conducted in the lab. But that's not always the case. As with everything we've been talking about, it depends on what your intentions are and what you're trying to get from the setup. So when a study takes place in the real world, it occurs in what we would call a field setting. And because it's occurring in the environment in which the behavior would naturally occur, you tend to have pretty high external validity. And specifically here, you're having high ecological validity because it's happening in the environment that it should happen in. But it's important to note that laboratory research can specifically be designed to be just as realistic, especially as the research conducted in the real world. So we can call this experimental realism, where you can control settings in the environment to be as close to the real world as possible. So you can choose tasks that are very similar to what's being done in the real world. You can feel real emotions in the lab if the situations are correct. We're just controlling for some external variables, and that isn't necessarily a bad thing. So even though field studies or research in the real world can be good because it has that high external validity, don't assume that lab studies don't have high validity, high external validity just because they're lab settings. That's not always the case. For our generalization mode, because the goal is to generalize, they care very, very much about external validity. So when conducting research where generalization is your goal, you need to have a representative sample. You need to go out of your way to have a sample that is representative of your population that you want to generalize to. And we also have to consider that ecological validity, how it applies to the real world, because you want to think about if your lab study actually could be applied to the real world. So is you're responding to playing cards as yes, I've seen it, or no, I haven't, lying or telling the truth, is that equivalent to people lying to the police or something like that? Maybe you have to do some extra work or some extra studies to make sure it's generalizable to other settings. Or maybe you need to pick a different method. Maybe by adding that motivation to lie is making it more closely applicable to a setting where people might lie in the real world. So you can critically think about these studies, and especially when you're designing them, you can start thinking about how ecologically valid is this setup in comparison to what they'd experience in the real world. And then lastly, when we're looking at our theory testing mode and the real world, so this is that idea that sometimes if you're testing a specific theory, you don't care if you can apply this to the real world. You don't care if you can apply this to a larger, more general population. Your goal is really just testing a theory. So in those cases, you decide that it isn't as important, and that's fine. And the textbook includes this sort of cute little summary of good ways to critically think about looking at popular media articles with this latest scientific breakthrough. And some of these questions apply for research articles as well, not just popular media. But instead of saying, look at this single interesting result, maybe critically think about the situation and ask, has it been replicated? That's a single study. What about the rest of the literature? And so these are all basically the ideas we've talked about throughout this chapter, and a lot of them throughout the whole course, so I won't subject you to reading that. This lecture is long enough already. I will leave it there. And that is it for the course. Next time I will interact with you will be for the final exam. So good luck with studying and have a great day.